Hello students. In today's module, we'll be learning about vesicular transport. Vesicular transport is a major cellular activity responsible for molecular traffic between a variety of specific membrane-enclosed compartments or organelles. The selectivity of such transport is therefore key to maintaining the functional organization of the cell. For example, lysosomal enzymes must be transported specifically from the Golgi apparatus to the lysosomes, not to the plasma membrane or to the endoplasmic reticulum. These proteins are transported within vesicles, so the specificity of transport is based on the selective packaging of the intended cargo into vesicles that recognize and fuse only with the appropriate target membrane. Because of the central importance of vesicular transport to the organization of eukaryotic cells, understanding the molecular mechanisms that control vesicle packaging, budding, and fusion is a major area of research in cell biology. So what is vesicular transport? Intracellular membrane systems do more for the cell than just provide increased membrane area. They create enclosed compartments that are separate from the cytosol, thus providing the cell with functionally specialized aqueous spaces. Because a lipid bilayer of organelle membrane is impermeable to most hydrophilic molecules, the membrane of each organelle must contain transport proteins that are responsible for the import and export of specific metabolites. Each organelle membrane must also have a mechanism for importing and incorporating into the organelle the specific proteins that make the organelle unique. All proteins begin being synthesized on ribosomes in the cytosol, except for the few that are synthesized on the ribosomes of mitochondria and plastids. There are three fundamentally different ways by which proteins move from one compartment to another. Number one, by gated transport. Number two, by transmembrane transport by membrane-bound protein translocators. And number three, by vesicular transport through transport vesicle. Newly synthesized proteins enter the biosynthetic secretory pathway in the endoplasmic reticulum by crossing the ER membrane from the cytosol. Subsequent transport from the endoplasmic reticulum to the Golgi apparatus and from the Golgi apparatus to the cell surface and elsewhere is mediated by transport vesicles, which transfer proteins from membrane to membrane or from lumen to lumen or to the extracellular space by cycles of vesicle budding and fusion. The pathway from the endoplasmic reticulum via the Golgi apparatus to the cell surface is often referred to as the default pathway because proteins seem not to require special signals to follow it. Any protein that enters the endoplasmic reticulum and folds and assembles properly will automatically be transported through the Golgi apparatus to the cell surface unless it contains signals that either detain it in an earlier compartment en route or divert it via the Golgi apparatus to lysosomes or secretory vesicles. Transport vesicles play a central role in the traffic of molecules between different membrane-enclosed compartments of the secretory pathway. Since vesicular transport plays a central role in the organization of eukaryotic cells, understanding the molecular mechanisms that control vesicle packaging, budding, and fusion is a major area of research in cell biology. Experimental approaches to understanding vesicular transport. Progress towards elucidating the molecular mechanisms of vesicular transport has been made by three distinct experimental approaches. Number one, by isolation of yeast mutants that are defective in protein transport and sorting. Number two, by reconstitution of vesicular transport in cell-free systems. And lastly, by biochemical analysis of synaptic vesicles, which are responsible for the regulated secretion of neurotransmitters by neurons. Each of these experimental systems has distinct advantages for understanding particular aspects of the transport process. Most important is the fact that results from all three of these avenues of investigation have indicated that similar molecular mechanisms regulate secretion in cells as different as yeast and mammalian neurons. Coat proteins and vesicle budding. The first step in vesicular transport is the formation of a vesicle by budding from the membrane. The cytoplasmic surfaces of transport vesicles are coated with proteins, and it appears to be the assembly of these protein coats that drives vesicle budding by distorting membrane conformation. The coat is thought to perform two principal functions. First, it concentrates specific membrane proteins in a specialized membrane patch that will then give rise to the vesicle membrane. So it helps select the right molecule for transport. Secondly, the assembly of coat proteins into curved basket-like lattices deforms the membrane patch and thereby molds the forming vesicles. Now, three kinds of coated vesicles have been characterized, and they appear to function in different types of vesicular transport. They are clathrin coated, COP1 coated, COP2 coated. Each type is used for different transport steps in the cell. Clathrin coated vesicles. Clathrin coated vesicles are responsible for the uptake of extracellular molecules from the plasma membrane by endocytosis, as well as the transport of molecules from the trans-Golgi network to lysosomes. The coats of clathrin coated vesicles are composed of two types of protein complexes, clathrin and adapter proteins, which assemble on the cytosolic side of the membranes. Adapter proteins or adaptins are required to bind the clathrin to the membrane and to trap various transmembrane proteins. Some of these transmembrane proteins act as receptors to capture proteins that are being transported inside the vesicle. We call them cargo molecules. In this way, a selected set of membrane proteins and the soluble cargo molecules that interact with them are packaged into each newly formed clathrin-coated transport vesicle. There are at least 4 types of adaptins, each specific for a different set of cargo receptors. Clathrin-coated vesicles budding off from different membranes use different adaptins and thus package different receptors and cargo molecules. Now let's look at COP-coated vesicles. 2 other types of coated vesicles have been identified as budding off from the endoplasmic reticulum and Golgi complex. These vesicles are called non-clathrin coated or COP-coated vesicles. COP stands for coat protein. There are 2 classes of COP-coated vesicles, COP1 and COP2 coated vesicles. COP2 coated vesicles bud from the endoplasmic reticulum and carry their cargo forward along the secretory pathway to the Golgi apparatus. In contrast, COP1 coated vesicles bud from the endoplasmic reticulum Golgi intermediate compartment or the Golgi apparatus and function in the retrieval pathways that serve to retain resident proteins in the Golgi and endoplasmic reticulum. The coats of COP1 and COP2 coated vesicles are composed of distinct protein complexes, which function analogously to collaterin and adapter proteins in vesicle budding. The assembly of vesicle coats also require GTP binding proteins, which appear to regulate the binding of coat proteins to the membrane. The budding of both clathrin-coated and COP1 coated vesicles from the Golgi complex requires a GTP binding protein called ARF, which stands for AGP ribosylation factor, while the budding of COP2 coated vesicles from the endoplasmic reticulum requires a distinct GTP binding protein called SAR1. The role of these proteins is illustrated by the function of ARF in assembly of COP1 coated vesicles. The first step in vesicle formation is the association of ARF bound to GDP with Golgi membrane. Proteins in the Golgi membrane then stimulate the exchange of GTP bound to ARF for GTP, and the COP1 coat proteins bind to the ARF GTP complex. Assembly of the coat is then followed by deformation of the membrane and vesicle budding. ARF then hydrolyzes its bound GTP, leading to the conversion of ARF to the GDP bound state and the dissociation of coat proteins from the vesicle membrane. Now let's look at vesicle fusion. The fusion of a transport vesicle with its target involves two types of events. First, the transport vesicle must specifically recognize the correct target membrane. For example, a vesicle carrying lysosomal enzymes has to deliver its cargo only to lysosome. Secondly, the vesicle and target membranes must fuse, thereby delivering the contents of the vesicle to the target organelle. Research over several years has led to the development of a model of vesicle fusion in which specific recognition between a vesicle and its target is mediated by interactions between unique pairs of transmembrane proteins, followed by fusion between the phospholipid bilayers of the vesicle and the target membranes. Proteins involved in vesicle fusion were initially identified in James Rothman's laboratory by biochemical analysis of reconstituted vesicular transport systems from mammalian cells. Analysis of the proteins involved in vesicle fusion in these systems led Rothman and his colleagues to propose a general model called the snare hypothesis, in which vesicle fusion is mediated by interactions between specific pairs of proteins called snares on the vesicle and target membranes. We call them V snares and T snares respectively. This hypothesis was supported by the identification of snares that were present on synaptic vesicles and by the finding of yeast secretion mutants that appeared to encode snares required for a variety of vesicle transport events. For example, transport from the endoplasmic reticulum to the Golgi in yeast requires specific snares that are located on both the vesicle and target membranes. The formation of complexes between V snares on the vesicle and T snares on the target membranes then leads to membrane fusion. There are at least 20 different snares in an animal cell, each associated with a particular membrane-enclosed organelle involved in the biosynthetic, secretory, or endocytic pathway. Rab proteins make an important contribution to the specificity of vesicular transport. They are monomeric GTPases with over 30 known members. Like the snares, each Rab protein has a characteristic distribution on cell membranes, and every cell organelle has at least one Rab protein on its cytosolic surface. These proteins are thought to facilitate and regulate the rate of vesicle docking and matching of V snares and T snares as required for membrane fusion. Once a transport vesicle has recognized its target membrane of an organelle and docked there, it unloads its cargo by membrane fusion, and thus the transport is completed. Vesicular transport is a major cellular activity responsible for molecular traffic between a variety of specific membrane-enclosed compartments. Vesicular transport helps to maintain the differences between these compartments by directed, selective transport of particular membrane components from one compartment to another. Transport vesicles bud off from specialized coated regions of donor membranes. The assembly of the coat, clathrin, COPE 1 and COPE 2, helps to collect specific membrane and soluble cargo molecules for transport and to drive the formation of the vesicle. And finally, snare proteins and GTpases called RABs guide vesicle transport, docking to target membrane, and mediate membrane fusion.